Assalamualaikum, my name is Nur Anissa binti Zainul from the Bachelor of Science Biology, Faculty of Applied Science, University Technology, Mara. Today, I will be presenting my final year project entitled Adaptive Physiological and Biochemical Response of Rice or Rhiza sativa to Drought Stress. Global climate changes has increased the air temperature and altered the rainfall pattern in Malaysia, resulting in more frequent and several drought events. These climate changes negatively affect agricultural productivity and threaten the national food security. As we know, rice is one of the most important staple food crop in Malaysia and play a significant role in ensuring national food security. However, rice is highly sensitive to the drought stress because it requires a continuous water supply throughout its growth. When water availability decreases, important physiological processes such as photosynthesis, nutrient uptake, and plant growth are negatively affected. To survive under drought conditions, rice plant active several defenses mechanisms. One of the most important biochemical response is the accumulation of proline, which function as the osmoprotectant by maintaining a cellular water balance and protecting a plant cell against the oxidative damage. Rice is the staple food in Malaysia, and our food security depends on consistent rice production. However, Malaysia still imports part of its rice supply, making the country vulnerable to the global disruption such as those experienced during the COVID-19 pandemic. Furthermore, climate changes has increased the frequency and severity of drought. According to Malaysian Meteorological Department 2026, several regions in Peninsular Malaysia experience severe to extremely dry conditions. Therefore, understanding how rice responds physiological and biochemical to drought stress is important for developing more drought-tolerant rice variety. This study consists of two objectives. The first objective was to evaluate the effect of drought stress on the plant height, dry weight, chlorophyll content, and relative water content. The second objective was to determine a proline accumulation as a biochemical indicator of drought tolerance. Moving on to the methodology. To ensure uniformity across all experimental units, seeds were first meticulously selected based on health and uniform length of approximately 1 cm. Next, the selected seed underwent inhibition by being soaked in distilled water for 24 hours. Following soaking, the seed were allowed to germinate in a single control container for 4 days. Once the consistent germination was observed, healthy seedlings were transplanted into 15 individual pods to establish our experimental unit for treatment application. This study evaluated 5 distinct water regimes to stimulate a varying degree of water stress. 100% water availability served as a control group. 75%, 50%, 25%, and 0% water level represent as mild, moderated, severe, and extreme drought stress treatment respectively. The best volume for 100% control water was calculated as 300 ml, which was determined through preliminary trial observation and the potting mix volume of the polybag used. Data collection is categorized according to the two main research objectives. For Objective 1, evaluate a morphological and physiological growth response by measuring a plant height, dry weight, chlorophyll content using a non-destructive spectrometer and a relative water content (RVC). While for Objective 2, focus on biochemical osmotic adaptation by quantifying a proline content using a UV spectrophotometer at its designated absorbance wavelength. Finally, all collected quantitative data were subjected to statistical analysis using ANOVA and post-hoc mean comparison tests to establish significant difference between treatment regimes. Figure 4.1 shows the effect of drought stress on the plant height. Plant height decreased with increasing in drought stress. Highest growth was observed under 100% water supply. No significant difference between 75% and 50% treatment. Plant height decreased progressively with increasing drought stress because water deficit reduced cell turgor pressure, limiting cell division and elongation. Consequently, shoot growth was significantly inhibited under severe drought conditions. Figure 4.2 shows the effect of drought stress on the dry weight. Dry weight decreased under drought stress because reduced water availability limited photosynthesis and carbon assimilation, resulting in lower biomass accumulation. Plants also allocated more energy towards survival rather than vegetative growth. Figure 4.3 shows the effect of drought stress on the chlorophyll content. Chlorophyll content remained relatively stable under moderated drought, which is 75% to 25% water supply, indicating that the photosynthetic pigment will still maintain. However, under severe drought, which is 0% water supply, chlorophyll degradation occurs due to oxidative damage and impair chloroplast function. The 4.4 shows the effect of drought stress on a relative water content (RVC). So relative water content decreased with increasing a drought stress. 100% and 75% water supply maintain high leaf hydration. Significant loss of water content occur at 25% and 0% water supply. Relative water content decrease with increasing drought stress because reduces soil moisture, lower leaf water status, and turgor pressure. Lower RVC reflect impaired water uptake and reduced physiological activity in rice plant. Figure 4.5 show the effect of drought stress on the proline content. So proline concentration increased with the drought severity. Highest accumulation occur at 25% water supply, while proline dropped to 0 at 0% water due to the tissue death and metabolic collapse. Proline accumulation increased as the drought stress intensified because proline functions as an osmoprotectant, helping to maintaining osmotic balance, stabilize protein and membrane, and scavenge reactive oxygen species. However, proline content declined to nearly zero under 0% water supply due to severe tissue damage and metabolic failure. So, for discussion for Objective 1, plant height decreased as a drought stress increased because water deficit reduced the cell turgor, limiting cell expansion and elongation. However, proline accumulated under moderated drought and act as an osmoprotectant. It maintained osmotic balance and cellular turgor, allowing continued cell growth under severe drought, and protective effect of proline was insufficient due to excessive dehydration, resulting in the lowest plant height. For dry weight, drought reduced dry weight because photosynthesis and carbon assimilation declined. Proline helps stabilize protein and enzyme involved in metabolism, allowing biomass production to continue under moderated drought. Under severe drought, metabolic activity declined, likely resulting in a lower dry weight. For chlorophyll content, although drought usually reduces chlorophyll, but chlorophyll content value remain relatively stable under moderated drought because proline acts as an antioxidant and it scavenge reactive oxygen species and protect chloroplast membrane from oxidative damage. Under 0% water supply, oxidative stress exceeds the protective capacity of proline, leading to chlorophyll degradation. Relative water content decreased because soil water become limited. However, accumulated proline function as an osmolyte by lowering a cellular osmotic potential, allowing cell to retain more water. This helps maintain higher relative water content under moderated drought. But under severe drought, the water loss exceeds the protective effect of proline. So, for proline accumulation is one of the major biochemical response of the plant to the drought stress. Besides functioning as an osmoprotectant, proline stabilizes protein and membrane while scavenging reactive oxygen species. This mechanism helps explain why plants under moderated drought maintain better growth, chlorophyll content, and relative water content compared to plants under severe drought. This mechanism shows how the proline accumulation responds to the drought. By osmotic adjustment, maintain cell turgor, scavenges reactive oxygen species, and stabilize protein and membrane. So the conclusion is the drought stress significantly reduced plant height, dry weight, and relative water content. Chlorophyll content remains stable under moderated drought but decline under severe drought. For objective 2, proline accumulation increased under moderated drought, indicating that osmotic adjustment, but proline declined under extreme drought because of the severe tissue damage. For future study, different Malaysian rice varieties should be compared to identify cultivar with better drought tolerance. In addition, antioxidant enzymes such as superoxide timotase, catalase, and ascorbic peroxidase could be analyzed alongside proline. Finally, a field trial and molecular study are recommended for further validated this finding. So that's conclude my presentation. Thank you very much for your attention.